Hi, my name is Pak Wong from Penn State Biomedical Engineering. In this video, we are going to talk about what are germs and how they are detected. What are germs? Germs are tiny organisms or microbes that can cause disease. There are many microbes everywhere in the environment, even in our body. However, not all of these microbes are harmful. Some of them can actually help us in digesting food and maintaining a balance of our body. However, some of them can cause diseases. Example of germs that cause diseases are bacteria, viruses, and fungi. So these organisms are so small, usually you can only see under a microscope. In this video, we're actually seeing some of the bacteria on a glass slide, and there's one silly one actually got stuck on the glass slide because right, it's kind of spinning on the surface. The body system that helps fight off sickness is called the immune system. In our immune system, there are two types of germ-fighting cells. They are called lymphocytes, who help us to remember some of these invaders. Another type is called phagocyte. So phagocyte are cells in our body that can engulf or eat some of these germs. So in this video, we're going to see a neutrophil, one of the phagocytes, that is in action is trying to catch the bacteria, and then it will engulf it. Got it. Since there are many different types of germs, it will be useful for the doctor to know the type of germs that cause the infection. For example, a doctor may prescribe an antibiotic to the patient if the doctor knows the infection is caused by bacteria. However, the same antibiotic will not be useful if the infection is caused by viruses or fungi. So even better, if the doctor knows the particular species of germs that cause the infection and whether they are resistant to the antibiotics. With that information, the doctor can be prescribed drugs that's much more precise and better and also avoid unnecessary treatment. Since the germs are so small, it is actually a very difficult task to identify them, to detect them, and know their species. So luckily, scientists and engineers have developed biosensing method based on the molecular signatures of these germs. So in all living organisms, their genetic material such as DNA and RNA that actually encode the protein required for the functions of these microbes, such as for their growth or maybe reproduction. So the genetic sequence of this DNA and RNA actually represent a unique and specific way for us to detect and identify these germs. How do we detect this DNA or RNA inside the germs that are so small? People have identified enzymes that can replicate or making copies of these DNA molecules. Bioengineers design machines that can repeatedly copy these DNA fragments. So, for example, one copy of this DNA can be replicated to two will become four, eight. The process are repeated until millions of copies of DNA are made. The presence of the amplified DNA can be detected by mixing color dyes that bind to the DNA. If there are germs, so it will be indicated by a large amount of light. In contrast, if there are low germs, the no DNA will be amplified and the amount of light will be very low. This process can be used to detect different types of germs such as bacteria and viruses. For example, SARS-CoV-2, the virus that caused COVID-19, can be detected by this method. If you want to learn more about germs and how they are detected, so please visit our website, like our Facebook page, and also check out our YouTube channel.